exactly. We're jumping into chapter 5. We got D Money on the last one. And we got Jen. She gives the peace signal. We can't see you. Yeah, we're all worn out, but we're finishing, and there's more printing going on now for some reason. I don't know why. Okay, so the first thing is to design an experiment. So some students really don't know how to do this. So the first thing is to create a hypothesis. It should be like an if-then, right? So let's say I said, if, I have an example, right? So if I give plant love in the form of long conversations, then they will grow more than a plant that is not. So now you have to have two different groups, the control group and the experimental group. The control group, they're treated normally. They're given the same amount of sun, the same amount of water, the same environment, the same seeds. But the only different thing is called the independent variable, the variable that you impose on it, which is the 30-minute conversations. The results of the experiment that you would collect is to see how much they grew. That's called the dependent variable. And they would either confirm or reject your hypothesis. Now, in the event that your hypothesis is proven not true, then you can't change your hypothesis, right? So we'll see that question come up in a second. All right, so this thing, so state a hypothesis to be tested. You could make any hot temperatures cause the seeds to die, or hot temperatures cause them to size of the population, they say, yeah, or cause the population to grow slower or something like that. And then what you would do is you would have a control group that would be in normal temperatures, one of them that would be in very hot or very cold, depending on what you set out to test, and then identify the type of data that would be collected, how many of them grew, how many of them. Yeah, all right, fine. In an experiment to determine the effect of exercise on pulse rate, a student checks his pulse rate before and after exercising for several minutes. The purpose of checking his pulse rate before, that's a control group. A student hypothesized that this one, so you can't change the hypothesis. It may lead to further investigation is the answer. A student hypothesized that drinking tea would cause an increase in pulse rate. He measured his pulse. He didn't measure it beforehand. One error with this hypothesis. What was the error with this one? I don't know. I didn't do this one. Different types of ticks and different numbers. What are you trying to test? Right, you can't test two things at once. Okay, good. Talking about students' proposed data will not likely test what the student intended. What did he intend to test? How well ticks survive hot water machine with detergent? They both have detergent. Something like that. I don't even get what he did. Identify one error of the student's design in the shaded area and explain how the student should change the experiment to correct the error.
Data to be collected, a number of ticks around the never mind.
Because the control group's not control.
It's not a control group, and also, what did he want to test? He wanted to test hot water. Right, fine, but he has completely different things. Oh, so the error with the hypothesis, can ticks survive hot water and detergent wash cycle in a washing machine?
Okay, the error with the hypothesis is that he's testing cold water.
And it's a question. It has to be a statement.
Oh, okay, got it.
Identify one. Oh, yeah, it's not if then.
It's not an if then, or it's a question, I think is the real fine. And then identify one error in the shaded area, how the student should change the experiment, right? So you'd have to use the same number and the same fine. This one here is a hard one because students are used to like the control group being 0 and then it being another extreme. But here the control group's the neutral one and you're testing both edges of it. So fine. And describe the type of data to be collected, fine. It's not hard. All right. This next one, fine, easy. Graphs, you got a graph. The first one I think is easy, you go by 10. The second one they have to go by 0.5. A lot of them will be very resistant to go by point. Yeah, 0.25 is even better. But a lot of them will be resistant to set the axes by, and also they'll say that you can't fit the full 2000, 2001 written out. Yes, you did 00, 01, 02. Also, yeah, also this you can't label. This is actually important. I thought this was somewhat here, Command D. You have to label, if you label this 0, right?
Yeah.
Oh, then you have to always go by that. So if this is 0 and then this is 2000, That's a mistake.
That's too far.
No, that's right. The next one would have to be 4,000. So you could have just started by having this year here be the 2,000 year. That's okay. But you couldn't start with 0 then 2,000. You got to go consistent. You understand? So even if some of them they'll want to start with like that, I think a lot of them know. But if you want to start with 1 here, well then you can't do 1.25 next. Now you have to have a consistent amount of growth.
What's 47?
47. Which statement could possibly explanation with a drop in mercury concentration in the fish of this between 2000 and
You need the graph.
2003? Yeah, I guess you'd have to have the graph. All right, fine. So these are all graphs. I think you should be able to figure out how to do them. This one here, you just see it sticks out. Gene 4 is the highest. That's the answer. This one here, you have temperature relates to male or female. In the, as the temperature changes, the probability of getting 1 is more, and it's 85%, 85 out of 100. I guess the answer to this one here is limiting factors, because temperature and rain could limit the amount of something that would grow. It's not going to be any of the other ones. And then 2 is the healthy glucose, right? That's homeostasis. Whereas the other ones, the only other option would be 3, I guess, but that just rose and got, they say 65 to 104 is the healthy range, so to have it be all the way up there.
It's 140 up there.
Right, not 104. Yeah, it does. True. Optimal temperature for enzyme. The enzyme responds, what? Answers 1. The enzyme responds in a similar way change in temperature. Fine. All right, fine. We don't have to go through all these. Checking these charts. We don't have to go through everything. And then you have the tools for measuring length, the volume. Fine. Here, if it zoomed in, you would be looking at 1. It zooms in most closely to the center, right?
Yeah.
For 44, what'd you get? What'd they use to obtain the better view?
I did. I did.
Stained it. Okay, fine. Wear goggles. This is chromatography. When you dip it in and you figure out what's.
Explain what chromatography is.
Yeah, I don't really know. You use this paper, and you dip it into the thing, and then I don't know. Based on what comes up on the thing, what type of. Yeah, it'll like move up based on whatever. Gel electrophoresis, I think they all know. You match it with the thing. Fine, that's easy. Okay, this one I didn't really explain it at all. I agree. Do you guys know how to do this?
Yeah, it's what we did before.
Right. So one reason for the change. So what happened in B? You put starch solution with enzyme Z in there. And then after an hour, starch solution with enzyme Z turned blue-black. Why did that happen?
Because osmosis, like it went through the starch indicator.
What went through the?
Starch indicator went in the bag.
Where did they put the starch indicator solution originally?
House, like here.
Oh, right. So the starch indicator solution fits through the bag, and it went in there.
That's for A.
For B also.
For B, the sugar's on the outside.
Right, but there's still starch solution in there. It's an indicator for starch, not only sugar. And how would the results have been different in beaker B if the enzyme digest protein was used instead of enzyme Z, an enzyme that digests protein? I don't know what's the answer to that one.
It needs that specific enzyme, otherwise it wouldn't digest the starch. I might be wrong.
Right, so what? It wouldn't digest the starch.
Enzyme Z digests the starch, whereas
this other one would not digest the starch.
I think.
I don't know. Okay, fine. And then what's this thing here? Is this similar at all to this or no?
This is like down.
State the effect of increasing the percent of salt in the solution used to water the plants on the average final height of the plants. So the more salt, the less likely it is to surprise.
Yeah, and if you put it in salt, the water will spill out and it'll shrink and die.
State one way diffusion was involved in the cause of death. Right, so the water diffused out because there was more salt outside and it got small. Fine. Fine. And then the next one here. Yeah, this one.
The tool?
There's no one tool using the beaks of labs. Explain why the special feature of this beak represented either an advantage or disadvantage to the competition. Yeah, the beaks of labs. Explain why the special feature of this beak represented either an advantage or disadvantage in the. I don't know. It has nothing to do with anything.
Oh yeah.
A what?
Like something specific.
We did it along. It was a clipped kind of thing, whatever it's called.
Like I'm sure the kids will know it. It's like obvious.
A clip?
It was like a.
Yeah, we did it in class.
Oh, okay, fine. And then what are they? And what is it? What's the advantage or disadvantage of the competition?
Little small
Oh,
one.
that's back to the small beak or
Yeah.
the large beak. Fine. All right, fine. And then what happened in this cell? Changes were caused from the transference of
Distilled water to salt water.
red onion to undergo the change. Right. So it shrunk. So it must have gone into salt water.
Great job.